Ma'am, good morning, ma'am.
Good morning. So I'll come back later at
Yes,
9:45.
ma'am.
Yeah. Okay.
Yes, ma'am.
Okay. 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 Thank you. I'll just unmute and close my video and go and come back. Yeah.
Yes, ma'am.
Yeah. Thank you. Thank you.
Sir, good morning, sir. Ramesh Babu. Just a second. Good morning, sir.
Good morning, sir. Good morning. Ramesh Babu, good morning. How are you?
Fine, sir.
Fine. Okay.
Yes, sir.
सर गुड मॉर्निंग।
Good morning.
जी सर, आपका एक ही नाम से कनेक्टेड है सर?
मैं बस एक दूसरे से जुड़ रहा हूँ।
जी सर।
Yes, connected. Thank you, Piyush.
Good morning to Honorable VC sir. Am I audible, sir?
Good morning. Good morning.
Sir, I think Madam has already appeared on the screen. And
Yeah. Good morning, Madam. Good morning.
Good morning, sir. Good morning.
How are you,
Very good
madam?
morning. I'm good, sir. How are you, sir?
Fine, fine. You are settled in Bangalore only.
Bangalore, sir. Yes, sir. Yes,
Very,
sir.
very nice. Very nice.
So nice to see you on the seat,
Thank
sir.
you, madam. Thank you. After a long time, even I left 1.5 years before ICR.
I know, sir. I used to come and meet you in the education
Yeah.
section.
Yes, madam. Yes, I remember.
Yeah.
Welcome,
Sir.
madam. Welcome. Most welcome.
Thank you, sir. It's an honor for me. Thank you.
Sir, I seek your kind permission to start the proceedings of the
Yeah,
webinar,
please. Please continue.
Thank you, sir. Good morning, everybody. Welcome to this online event, which is being organized by Department of Entomology of Maharana Pratap University of Agriculture and Technology, Udaipur. It's indeed a matter of great pleasure that Madam Chandish Balal is with us. And of course, we have got with us Honorable Vice Chancellor, sir, our patron and our mentor. Of course, this webinar is entitled as a promise of biological control for sustainable pest management. In fact, madam, since the beginning of the pandemic way back in March 2020, under the leadership and under the guidance, under the patronage of Honorable Vice Chancellor, sir, this university is consistently organizing webinars and with a person of eminence like you and many others. And he has instructed us to have very high-rated people and the people who are who are the legend of their own discipline. And of course, from last 1 month, we were in the process as to how we can plan some webinar in the Department of Entomology, particularly in the domain of biological control. And it was so heartening that you have accepted our proposal. So at this outset, the university welcomes Madam Balal. Of course, we welcome our Honorable Vice Chancellor, sir. We welcome all our deans and directors. We welcome all our students, and we welcome all the participants. And right now, madam, we have got 928 participants on their laptops or on their mobiles, and they are eager to hear you. But at the outset, we request our Dean, Rajasthan College of Agriculture, Dr. Dilip Singh, to welcome the guests and to put forth the preface of this webinar. It's over to you, Dr. Dilip Singh ji, Dean RC.
Very good morning to all of you. Thank you, Dr. Nepalia, sir. Honorable Vice Chancellor, Dr. Narendra Singh ji Rathod Sahib, distinguished speaker and former director ICAR NBAIR Bangalore, Dr. Madam Chandish Balal, Dean CTA Dr. Ajay Sharma, Director Research Dr. SK Sharma, our registrars, controller, my fellow dean, directors, head of departments, faculty members, scientists, students, and dear participants. I extend a warm greeting to all of you on joining us in this virtual event organized by Department of Entomology of our college. I feel privileged to accord warm welcome to the distinguished guest and all of you in this national webinar on promise of biological control for sustainable pest management, organized under direction and support of Honorable Vice Chancellor, sir. We are overjoyed to offer a warm welcome to our patron and chief guest, Dr. Narendra Singh ji Rathod, Honorable Vice Chancellor, in this webinar. Sir, under your guidance, our different departments have created opportunity to knowledge enhancement on issue of vital importance during this disgusting pandemic time by way of virtually inviting senior subject expert to discuss subject of present day requirement. Sir, your far-sighted visions and quest of bringing about excellence in academic is very well evident from conduct of almost 100 international and national webinars in last 1 year. And today is the turn of discussion on promise of biological control for sustainable pest management. We express sincere gratitude to Honorable Vice Chancellor, sir, for having given his kind-hearted permissions and patronage for this event. Sir, we extend a warm greeting to your honor on your virtual arrival in this webinar.
Thank you.
We
Thank
offer
you.
a rousing welcome to our eminent speaker, Dr. Madam Chandis Balal, former Director ICAR, National Bureau of Agriculture Insect Resource and Project Coordinator, AICRP on biological control. Her assignment prior to superannuation exactly 1 year back is fair indication of her authority in the field of environment, safety and sustainability through biological pest management. Madam, by agreeing to our request in one pass to deliberate on this all-important subject has touched our heart. We extend a very warm welcome to your virtual arrival in MPUAT. I also welcome our registrar, Mrs. Kavita Pathak, Comptroller, Mrs. Manjubala Jain, all the deans, directors, head of department, faculty members, and participants in this important national webinar. Ladies and gentlemen, let me put before you a brief permeable with which we have articulated today's session. Insect pest disease and weed inflicts enormous losses to the potential agriculture production. With the start of Green Revolution in India, chemical substances were used extensively in the mission to raise food production in order to feed rising population. However, excessive use of agrochemicals, including insecticides, lead to environmental pollution, contamination of food and water, and development of resistance to chemical pesticides in several pests, which became major concern in agriculture production and sustainable pest management. There is a rising public concern about the potential adverse effect of chemical pesticides on the human health, environment, and biodiversity. These negative externalities, though cannot be eliminated altogether, their intensity can be minimized through development, disseminations, and promotion of alternative technologies such as biological and bioagent, as well as good agronomic practices, rather depending solely on chemical pesticides. That is where biological pest management is seen as an opportunity. As we all know, biological control is use of living organisms to suppress pest population, making them less damaging than they would otherwise be. Natural enemies of insect plays an important role in limiting the density of potential pests. As far as we know, there are 3 strategies for biological control of insects. The first one is importation, that is classical biological control. Second one is augmentation, and third is conservation and natural enemies. The specific technology within these approaches are constantly being developed and adapted to meet the changing need of pest management. The use of trichogramma against lepidopond insect is well known to farmers. The term like predators, parasitoids, pathogens, and competitors are widely known. Predators are mainly free-living species that directly consume a large number of prey during their whole lifetime, whereas parasites is an organism that live and feed on a larger host. Insect parasites are more precisely called parasitoids. Pathogenic microorganisms include bacteria, fungi, and virus. Entomorphopathogenic nematodes are mass-produced for use of biopesticide using in vivo or in vitro methods. Lots of research findings are still searching headway to farmer fields, for which we also need sound backing of technology outreach apart from continuing research. Biological control is an important component of integrated pest management, but it is for sure that use of biological agent can reduce the chemical pesticide usage and enhance sustainability. Through the AICR program, biocontrol, new approaches have been designed, and the biocontrol technologies have been improved, yet there is need to be fine-tuned and field tested for wide acceptance by the end of users. However, the effective planning of biological pest management with the focus on sustainability will need assessment of available resources, economic feasibility, economics of use, social factors, and the government policies. I hope that today's lectures by Dr. Balal will touch all aspects of biological control in reference to sustainable pest management. We are also sure to receive a handful guidance from our Honorable Vice Chancellor, Dr. Narendra Singh Rathod Sahib, to shape our future activities in this direction. With this preface, I once again extend a warm welcome to our Chief Guest and Honorable Vice Chancellor, Dr. Narendra Singh Rathod Sahib, eminent speaker, esteemed Dr. Chandis Balal, all the senior officers, deans, directors, head of department, faculty members, dear students, and participating scientists, and wish the webinar all success. Thank you very much, sir. Dr. Nepalia, sir, please.
Thank you, Dr. Dilip Singh, sir, for those words of welcome and for putting forth the preface under which this webinar is being organized today here in MPUAT. Thank you very much. Before inviting Honorable Vice Chancellor, sir, and our patron for his address, for the benefit of all those, because more than 900 participants from all over the country have joined, and our Honorable Vice Chancellor is very widely known, as very popular amongst all the scientists and all the scholars. But still, for the benefit of some of those students from outside MPUAT who may still not be knowing that much about Honorable Vice Chancellor, let me briefly introduce Honorable VC Sir to all of them. Dear participants, Honorable Vice Chancellor Dr. Narendra Singh Rathod comes from a very small village of district Pali of Rajasthan. And after very early struggle and after early school education in Udaipur, he then completed his graduation from College of Technology and Engineering in Udaipur. He then went out to the folds of IIT in New Delhi, from where he completed his MTech, and then he did his PhD as well. He's a true symbol of teaching, research, and extension. He came to the university services way back and then served this university in various capacities. And he here remained in a lot of administrative positions as well. He was the dean of 2 colleges here: College of Technology and Engineering, College of Food Science and Dairy Technology. He was student welfare officer, and then realizing his potential, he was appointed as DDG of engineering in ICR. But very shortly, government of Rajasthan snatched back him from ICR, and he was appointed as the founder vice chancellor of SKN University at Agriculture University at Jobner. And after successfully completing that term, he again went back to the folds of ICR, and he was a deputy DG of Education Division. And all of us know, all who are connected directly or indirectly with education in agriculture are connected. I think everybody knows as to what quantum reforms did he bring out during his tenure as DDG. He was responsible for implementing the 5th Dean Committee recommendation, and within that recommendation, the all-important Student Ready program was introduced by Dr. N.S. Rathod sir. Of course, he is so dynamic. When he came back to the university a year and a half back as Vice Chancellor of MPUAT, he very soon realized his presence here. Our university, which was struggling for its position at national level, it was a decimal 51st position, madam, when Dr. Rathod joined here. And within a year time, he could muster everything here, and our university rose to the position number 26 last year. And we are sure that with the consistent efforts of Honorable Vice Chancellor sir, whenever we meet next, you will find that there is another quantum jump in the position of our university. Most of his period as Vice Chancellor has been under the lockdown period and under the pandemic period. And during this, he was instrumental in guiding us for all the online classes, very timely completion of online examination, online evaluation of answer sheets, declaration of results, and award of degrees. And even online convocation was held on 24th of December 2020, which was very widely appreciated, because in that convocation, we could award a few degrees using mixed reality. And it was appreciated by Honorable Governor of Rajasthan and everybody in India. Sir himself is a prolific writer. He has written, I think you know that he's a prolific writer. He has written a number of books and number of research papers previously. Even during this lockdown period, he has written 4 books. He has authored 4 books, 3 of which were released by Honorable Governor sir on the day of convocation itself. So this speaks of the volume of leadership that he's providing. And because of his consistently writing, our faculty has been able to publish more than 400 e-manuals and e-compendiums for the quick subject matter delivery to the student during this last 1 year of pandemic period. Madam, He is very concerned about the research output, and our H index, which was also lagging behind, is now hovering around 52, our university H index. And as I was telling you, in the initial period, we have had 100, almost 100 webinars during this last 1 year and 2 months. So if you go by mathematics, it comes out to be one webinar per 4 or 5 days, I think, online event. So that's the greatness of our Honorable Vice Chancellor. He has got number of awards to his baggie, the latest one being the Lifetime Achievement Award by Aligarh Muslim University about 2-3 months back. And he has commercialized number of technologies. He's member of various bodies, accreditation boards. He's a man of great acumen and multifaceted qualities. I think if I wish to speak, I can keep on speaking about Honorable Vice Chancellor for hours together, but time is a constraint. So with these few words and with the gratitude to Honorable Vice Chancellor, sir, with great honor and respect, I invite Honorable Vice Chancellor, sir, for his address in this webinar. It's over to you, sir.
Good morning to one and all. Thank you very much, Dr. Virendra Nepalia ji. At the outset, I would like to take this opportunity to welcome our Madam Balal, our former director of ICR. I have seen her very closely when she was working there, and really she deserves appreciation. And the other day, when our head of the department, Dr. Manoj Mehla, came to me and he discussed the issue with Dr. Virendra Nepalia as well. And when your name came in our mind, we immediately decided to request you. And we are really grateful that you have accepted our proposal. And thank you very much. And we are eagerly waiting that how the things will materialize in this 1.5 hour. Thank you very much, madam. And at this stage, I am also thankful to my all deans and director, Dean of Rajasthan College of Agriculture, head of the Department of Entomology Department, Director Research, Director Extension, Dean CTA, Dean of Community and Science, and Dean of our Agriculture of Bhilwara as well. And all our colleagues from Senior Officer Council of a university, and our registrar madam and comptroller madam. And today, no doubt, this is a very interesting topic is selected that today we are going to discuss about different biological control which are sustainable as far as the pest management is concerned. However, in present context, there is a need to discuss that what is a sustainable agriculture, because everybody is talking about that we are in need of at least of 8.5 billion tons of a grain production across the globe by 2030, and that's why we will have to work extensively. But only problem is that a pest like insect, pathogens and weeds, they are estimated to cause a loss of approximately of a 27 in between a 27 to 42%. That is the statistics available at the national level. And that's why there is a need to have a new way out so that we can control this one. And it is further estimated that in case if we will not be having a right protection of a crop, especially with a right type of a pest and insect control, then this estimate loss will be extended up to the level of 50% or 50 to 75%. That is a estimate that people have made. So, and even people are expecting that there is a need to have a more than 15 times to improve 15 to 20 times to improve the use of pesticides and insecticide and other things in order to meet out this requirement. But basically, if we are using a synthetic pesticide, there are a lot of losses. There are a lot of problems there, and definitely madam will discuss. And today we are going to discuss that what is the sustainable agriculture and how we can decide an environmental friendly production system as compared to our traditional system, what our dean has also mentioned. And even we will have to see that what is a social sustainability, what is economical sustainability, what is a environmental sustainability. And in present context, when our country and likewise our country, every country is facing a problem of a production, productivity and profitability. At the same time, we are also facing a problem of quality production. And last year statistics was that around 141 kg of chemical fertilizers and insecticide and pesticide per hectare wise on an average we were using in our country. Although our position is third across the globe, but basically it insisted that as far as the quality production is concerned, we are lagging with the many of the countries. So that's why there is a need to have a biological control. And why this biological control is an environmentally friendly technology, that we will have to discuss. And even prior to biological control, let us see that what are the different principles of pest control and how we can have a pest control method which are available in. And when we are talking about sustainable pest control method, integrated pest control method is coming into the picture. So we will have to see that how we biological control method can also be integrated with the other methods, so that at least we will be get rid of a sum of amount of a insecticide and pesticide. But there other picture is also available. People are saying there are a lot of methods are available for biological control. We have got a number of advantages, but people are also highlighting some of the disadvantages as well. I will also give some of the input to our participant. And now question arises that how biological control affect our environment. That is one has to research. And basically we will have to research in the way that how we can increase the efficacy of this biological control method as well. Because what is happening, biological control, it has also been observed that they does not work as per our expectation, and even they are not popular amongst our different stakeholders, and there are number of risks are involved as far as the different options are concerned. So it is must to find out whether biological control is effective in our operation, and at the same time whether it is enough to control the pest and disease in isolation. If it is integrated, then it is good. So that's why we will have to see that what is its effectiveness and what is its overall visibility. So there are lot of things are available whenever we are discussing that our global population is increasing. As I mentioned that by 2030, 8.5 billion tons of a grain production across the country. And that's why we will have to see that how to increase the need of economic food production. And at the same time, there is also need to how to reduce the adverse environmental effect, because already whatever natural sources are available to us, like soil, water, everything has been degraded. And now when we are counting our organic carbon content in the soil, or even microbiological count in the soil, it is quite inferior. And at the same time, the water which is available, it is also not potable. So that's why we will have to see that how we can attend a sustainable agriculture. And whenever any scientific work comes into the picture, there are three aspects which are emerging. Very first aspect is a sustainability. We say how we can maintain the same supply in future as per our need. And second thing is that it should not damage to the environment and to our future. That is basically a scientific work is in need of that type of work. And third thing is that we should have excellence in the nature as well. So these are the three important things, and that's why in present context, whenever we are saying that we should not compromise the ability of future generation to meet their own demand, and then that's why we are always are coming forward to have some biological control method, especially for control of this insect and pest. And which is basically this, whenever we are talking about sustainable agriculture, it is not only giving that we should have an optimum supply, but at least it is a necessary component for our social sustainability. Even it is a necessary component for economical sustainability or environmental sustainability. So we will have to see that a sustainable agriculture and sustainable food system, how they are correlated, and especially biological control method, how it can be useful for our sustainable agriculture. And there are a lot of things are there. I don't want to go in details, especially what are the adverse effect on environment, especially because of synthetic pesticide, how we can minimize it. But people are expecting that with the help of a biological control, it is not only a sustainable and cheapest and most environmental safe pest control method, but at the same time it will provide additional benefits for the plant breeders as well as the consumer compared to the chemical control. So that's why we will have to see that in totality that how it can be closely linked to in a sustainable agriculture. And whenever we are talking about as far as the pest control is concerned, there are three words which are coming in my mind. First thing is that immediately we come to a word so-called prevention, means keeping a pest away from a becoming a major problem. So that is a first step which is generally is being recognized, that we will have to prevent our field from this possible attack of the pest and insects and other diseases. And second thing is that immediately there is a second word, so superation, that is how to reduce the pest number or how to reduce damage. Even reducing the damage to a acceptable limit, it is a permissible. People are saying that we can't remove 70% of the damage because of the pest and this, but we will have to see the third word which is immediately coming, that is a eradication. Eradication means how you can destroy the entire pest population. That is basically a sort of a pest principle which is available, and there are a lot of methods are available. I don't want to go deeply, but just for information to participant, that a physical method, this is one of the natural based method, and it is a non-chemical method, but at the same time second which is more dominating at everywhere in almost in all 28 states and 8 union territory in our country, that is a chemical method of pest control, but it is a highly dangerous, looking to its a gradual effect and even in its effect on the quality of our crop. And third method is a cultural method of a pest control. And of course, fourth one, which is today we are going to discuss, and Madam Balal will also throw a new light that how we can have a sustainable pest control method. In a sustainable pest control method, as I mentioned, it is only that a integrated pest management strategy has to be decided. And we will have to see that how optimally we can use cultural, mechanical, physical, biological, and chemical method along with our basically main emphasis should be there on a biological, so that we can have some practices to control the pest. And that's why in a IPM module, whenever we are deciding a IPM module, a very first point is that let us decide a some threshold limit, that let us set a action threshold. And before taking any pest control action, we will have to decide that at point at which the pest population and environmental condition will be will need a right type of pest control action, that we will have to decide. And then we will continuously, the second point and in strategic planning for IPM is that we will have a right type of action and identify a pest which is available, and then of course a right prevention and control method is there. So basically when we are comparing, when we are seeing a biological control, as the name indicates, it gives an impression that is the use of some living organism to control pest. And as a result of this, it may be a parasite or predators or disease organism which is introduced into the environment of a pest. And as a result of this, it will multiply in numbers, and finally it becomes more effective in reducing the number of pest organisms. The same idea is being utilized in a vaccination for COVID. Here in COVID as well, the sum of the amount of some virus is introduced in our body, so that our immunity will be utilized to boost to make some antibodies, so that we will be get rid of this problem. Same thing is that in a biological control, we will have to identify a right type of living organism, and we will have to make a right type of environment with our place where there is already some contamination is there, and then we will have to increase the this pest which we have introduced to multiply in numbers, become more effective in so that's why it is known as it is more sustainable in nature. And it is, as I mentioned, that it is also economical. And interesting thing is that which anybody can predict, that it has got a less legal and environmental implication as well. Otherwise, most of the chemical fertilizer and synthetic fertilizers, there are a lot of legal things are there. Day before yesterday, our former vice chancellor of our university, Dr. S.S. Chahal, has also published a news in a leading Times of India, where he has thrown that how we can control the pest and other thing. And he mentioned that in Punjab government has already banned some of the chemical fertilizers and even pesticides, especially in a basmati rice, which is basic idea is to get a more revenue for after educate import export. So that's why there is a need to have some biological control. I will share this complete newspaper clipping to madam as well, and he has given a very good thing that how we can have a some parasitics and how we can trap it just like aphids to control the aphids. Aphids basically it is a pest of plant which cause a larger damage to the plant itself, and essential nutrients are also being removed because of these aphids. So in order to control this aphids, a parasite webs is helps which ultimately lays eggs in the aphids and it destroy the aphids. Same thing we will have to see that a biological control method which we will have to see that how application of biocontrol agent safer to the environment and person who is applying this. And we will have, as I mentioned earlier, that there are certain disadvantages as well, and we will have to see it. And still there is a need to do some extensive research that people are saying it is a slow process. How we can expedite it? That is a major thing. Second thing is that people are saying that it is not responsible to completely wipe out a pest, and that's why we will have a people are saying it is not right choice, but we can supplement some of the pesticide or a requirement with the help of a biological control measure. So that's why there is a need to integrate it with the existing system, so that at least of to certain extent we will be get rid of a chemical insecticide and other things. And while it is cheaper in a long run, people are saying it is cheaper in long run, but at initial inception when we are setting up a biological control system, it is a costly endeavor. So that's why we will have to commend it, and we will have to do extensive research that whether it can be fickle in nature, because people are saying that it is a fickle in nature, especially biological control. And that's why even people are saying that a biological control methods, in case if adequate attention is not being provided, then it may affect the environment. I don't know how far people perception it may be of a you can say a thing which need a right type of deliberation, because what is happening, this has failed to control the original pest, and as a result of this it will enhance a targeted pest. And that's why people are saying that it is not working as per our expectation, because not implementing a reliable scouting program, even it is not conducting a quality assessment and not realizing enough natural enemies. There are a lot of myth is there. I can use the words precisely. These are the certain myths, and that's why we will have to go extensively that why it is not being so popular in present context as well. Because people are saying, especially in horticulture, in open field and orchard, this is not working effectively, because in orchard, in most of the horticulture crop as well, you can't control the complete environment, which includes your rainfall, it is humidity, temperature, which cannot be controlled. So it, because of this, there will be irregular growth of pest. So basically crop is more open to a range of pest, and that's why it is not so effectively working biological control. So it may prevent damage of a specific biological control as well. And that's why we will have to see that what are the direct and indirect attack of biological control agent on a non-targeted crop. Even what were the what is a dispersal of a bio control agent to a new area, and how you can stop this dispersal as well. And self motivated, or because of its own autonomously it is moving from one place to second place, and even because of interference of a human assistant as well, there is a chances that a biocontrol agents will move to a new area, which will not effectively contribute to the your original area. And there is also things which has been coming into number of papers that there is a change relationship between a controlled agent and a native species. That's why we will have to see its effectiveness as well, because if we see that recommended release dose of a trichoderma is also in a vegetables and field crop is it vary up to the 1 lakh per acre per week depending on level of pest infestation. So that's why classical biological control, they are not always working up to our expectation, because it is usually most effective against exotic pests, not and very less, especially on native insect pests. I don't know. Our Madam Balal will give some more insight as far as that how far they are effective, and at the same time. We will have to see that whether biological control method is alone is sufficient to control pest and disease, whether we will have some extensive research on this side, and can we get some more information, and can we identify some simple, convenient and efficient biological control devices, and which will definitely play an important role and greater role in the sustainable control of disease and pest in the future. But let us see that how we can integrate it, and what are the different options available in present context, and how far they are available as far as to get rid of the larger number of diseases and pests in agriculture, and especially in horticulture as well, in the forestry as well. We are not only confined on agriculture, but we will have to see in a totality that how it is effectively working in horticulture and forestry. Unfortunately, this is my observation. Or in a horticulture field, neither a right type of mechanization is available, and at the same time, as far as the insecticide and pesticide use is concerned, it is also quite cumbersome looking to the total vegetative growth of the most of the plant in the orchard. And at the same time, if we are going for any of biological control method, even it is not so suitable, and even people are trying their best to get some right solution for horticulture crop, because horticulture is also contributing 30% of our agriculture GDP as well. So that's why we will have to see that in a totality how biological control method can provide a promising option in our sustainable pest management. Even we will have to see that how it is effectively useful for a sustainable agriculture. So I'm sure that Madam Lal will throw a more light on this issue. As far as my knowledge is concerned, I can see that there are a lot of myth available in front of a biological control measures as well. But we will have to solve this problem, and we will have to see that that how socially, economically, and environmentally this biological control method is closely linked in a sustainable agriculture, and we should comment that how efficiently we can trap this biological control method, and even what will be the its future scenario as far as the IPM module is concerned. That is also perhaps we will have to deliberate on this issue. So there are a lot of researchable issues are available in front of this emerging area, and at the same time it gives an impression that it is a, as I mentioned earlier as well, that it is a economical, reliable and environmental friendly method, and by using a number of living organisms to reduce the pest population, but only with a certain the right type of a guide, and even we will have to create a right type of awareness in the mindset of a different stakeholder, not to only to farmers and grower, even to the this synthetics pesticides manufacturer as well, so that they should not spoil our agriculture. And even we will have to awaken the policymakers so that they will divert their attention, and for the want of a right type of a thing, and we will have to broaden our scope of work. That is my view on this issue. We will have to see that how we can meet the exact demand of this, and how effectively and how we can increase its efficacy in the use, and how we can control it. And even there is a need to have a collaboration and cooperation, and even a right type of networking is required at national level for a different scientist and even a different organization, so that they will contribute logically on different aspect of a biological control. And of course, in a local language, there is a need to have some good literature and how we can disseminate this technology. Today, our Director of Extension might have also joined, and some of our KVK people might have also joined. So on a mission mode, we will have to take this and how we can implement this particular procedure to control our pest problem. And that is a need of our. With these words, once again, thank you very much, Madam Malal, for sparing your valuable time. And thanks to all our participants. Already more than 950 participants has joined. And Dr. Mela has informed me that more than 2,000 people, student and participant and teachers has registered and continuously, and they have now may say close the entry, and otherwise this will collapse. And that's why they restricted not to enter more than that of a 1,000 participant at the time. But looking to the topic and seeing that madam you are coming, that's why everybody was so excited and people have joined like anything. So with this word, thank you very much, madam, and thanks to all organizers and our director, Director Extension, Dr. Virendra Nepali, and all participants. Thank you very much. Thanks a lot.
Thank you, sir. Thank you very much for your nice words and setting forth the tone of the webinar and the tone of the topic today. Now, much awaited lecture from Balal. But before that, as per protocol, let me introduce Madam Balal to our house, to all our students, because Madam, there are many young students from MPUAT and other state universities also participating, and they should definitely be inspired by your profile, I think. So friends, Madam Chandish Balal, she obtained her undergraduate and postgraduate degree, and of course the MPhil degree from Calicut University, and later on she did her PhD from the Mysore University. And she was a merit scholarship holder all throughout her educational career. So you can underline these words that she was a merit holder all throughout her educational career. She was recipient of merit scholarship, and her first posting as scientist was in ICAR, Indian Institute of Horticulture Research, way back in 1985. And then later she moved on to Project Directorate of Biological Control during the formative years of that particular institute. And then later she moved to the Project Directorate of Biological Control during the formative years, of course, which is now upgraded as ICAR, National Bureau of Agricultural Insect Resources. She was the head of Division of Insect Ecology from 2013 to 2016. And then she rose to the position of director of that institute from 2017 till her superannuation exactly 1 year back, as Dr. Dilip Singh was saying. She has always laid emphasis on environmental safety and sustainability by focusing her research efforts on conservation of biodiversity and biological control, which is of course a non-chemical mode of insect pest management. She has standardized and effectively she has gone under the production technologies for beneficial insects, which are being widely adopted by the commercial units, by the researchers, and by the scholars in their laboratories as well. She holds the largest, of course, the largest repository in Asia. She has 127 insect cultures with her. The prompt and continuous supply of quality live insect cultures made and production protocols developed by her are, of course, valuable services to the students community, biocontrol researchers, and commercial people as well. By interacting with farmers and conducting demonstrations trials on farmers' field, she has created confidence in the farmers on the non-chemical mode of pest management. Of course, she has handled more than 40 research projects. Our honorable sir is always of the opinion that one who holds a number of research projects in her hand is always a leading research scientist. And sir, madam holds more than 40 research projects she has handled. She has more than 300 research papers published in international and national journals and technical bulletins and symposium papers and all those. She has received international travel grant from DST, from IOBC, from CSIR, from CABI, from ICAR, from FAO, and Beijing Academy of Sciences. And she has presented her work in international conferences in many of the countries like Greece, China, Sri Lanka, Nepal, Bangkok, and USA, so on and so forth. She is, of course, an elected fellow of various professional societies and is a recipient of several awards as well, several recognitions. To name a few of them here includes T.N. Ananta Krishna Award, Dr. Sithanand Award, NBARI Scientific Excellence Award, Dr. S.P. Singh Biocontrol Lifetime Achievement Award, of course, the ICAR Punjab Rao Deshmukh Outstanding Women Agricultural Scientist Award, and very recently, in last couple of years, she has received 2-3 more awards like S. Pradhan Memorial Award from IARI and Prem Dhureja Endowment Award from National Academy of Agricultural Sciences, and of course, Dr. B.V. Dave Lifetime Achievement Award. And all throughout her career, she has been training the stakeholders. She has been emphasizing the stakeholders about the biological control, bioagents, and she has made concentrated efforts to create awareness amongst the masses on the importance of healthy environment and to popularize the concept of biological control. And this is, of course, all the stakeholders like students, the researchers, the industrialists. So these were a few words about Madam Balal. I think these are sufficient to inspire our budding entomologists, the budding agricultural scientists, the budding students. Madam, with these few words, may I, on behalf of Honorable Vice Chancellor, on behalf of the university, request you with folded hands to kindly deliberate on the issue which is to be discussed today. It's over to you, Madam Balal.
Am I audible? Am I
Yes, ma'am.
audible, sir? Okay. Is
Yes,
my
yes,
screen
madam.
visible?
Yes, madam. Yes.
Okay, okay. Good morning, Honorable VC Sir, Dr. Rathod, Dr. Nepalia, Dr. Dilip Singh, Dr. Ajay Sharma, Dr. S.K. Sharma, Dr. Mehla, Dr. Meenu Srivastava, Dr. Ramesh Babu, all the senior officials of MPUAT, and all the young researchers and students from MPUAT and outside MPUAT, and all the participants. I'm truly overwhelmed by the number of participants who are here today. A very good morning to you. I think I really consider this a great honor to be here speaking on my favorite topic, that is on biological control. And also, I think VC Sir's introduction has made it a greater challenge for me to address this topic. He has thrown several challenges right before I started my presentation, which makes my topic more challenging, my talk more challenging. And also thank you, Dr. Nepalia, for that very kind introduction about me. I do not know if I deserve that. First and foremost, I would like to say that through this lecture, what I am intending to do is, I would like to introduce the topic by giving practical examples and by giving global examples and Indian examples. And I should emphasize that this topic was prepared primarily keeping the young researchers and students in mind, mainly because I feel biological control can succeed only if I can convince the teachers to take more projects on biological control, if students take more biological control topics for their dissertations, if the policy makers understand the concept behind biological control and encourage the science of biological control, and if there are youngsters who are ready to take up entrepreneurship on production of biocontrol agents, if I can inspire all these people, I think my job of giving a talk here is 90% completed. So with this, I would start my presentation, and I have great memories about this university due to various reasons. One is because it is MPUAT is one of the centers of our AICRP biocontrol. So I have an emotional attachment to this university. And also I should say one of the earlier professors, Dr. Bajpayee, was with me for about 6 months on a project on biological control way back in late 1990s. And it ended up with an international beautiful publication. So I'm remembering that Dr. Bajpayee, I think he's a dean of Banda University right now. So with this, let me carry on with this topic. So the topic is promise of biological control for sustainable pest management. Now, what is sustainable pest management? I think the VC was also wanting to understand more about the sustainable pest management. Through sustainable pest management, what we are focusing is that we should be able to meet the current and future need of protecting human health and environment. So the concern about future of environment is what sustainable pest management looks at. It doesn't say don't use pesticides. It says judicious use of pesticides, because through sustainable pest management, we are supposed to ensure that our natural resources are safe and they are utilized efficiently, and they are there for the future generations. We don't destroy them now. We have to keep them for future generations. And another important point is that it should be economically viable. So how do I say biological control can be a promise for sustainable pest management? It's because biological control is ecologically sound. So we are ensuring that the future generations are getting the resources. Second one is that it is economically viable. And third point, socially just and adaptable. Even farmers can understand if we go out to them and try to convince them. Most importantly, I think each human being right now wants to make sure that his or her food is free of chemical pesticides. Now, whatever qualities I mentioned about sustainable pest management, I am sure that chemical pesticides would not be able to completely do that. So it is biological control which can to some extent respond to the requirements of sustainable pest management. Now, this is again for the students and the young researchers. What is the importance of taxonomy in pest management? First and foremost, we should be able to differentiate between the pests and natural enemies. See, generally, when we speak of insects, a common man says and looks at an insect only as a pest. But taxonomy helps us to differentiate between which is the pest and which is the useful insect. And also these days, you might have seen a lot of strange alien insects are entering our country from the borders. Now, to identify that, taxonomy is important. Another thing, when the invasives are entering our country, sometimes it enters because they are massive flyers. They can come through any source. Sometimes they are entering from our airports. They are entering from the different ports of entry. So these, suppose a consignment is coming, let it be a vegetable or a fruit, if it contains a pest, there should be sufficient taxonomic knowledge to identify that invasive and prevent the consignment or the pest from entering our country. For this, the quarantine agency and the plant protection officials should have some amount of knowledge on taxonomy. So insect taxonomy is very important for biological control of pests. Now, I'm just showing you one example of a museum, which is important for taxonomy. This is our NBAIR museum in Bangalore. And there are many such museums in IARI. I think MPUAT, you have a great museum. Now, my only request over here is most students can take up taxonomic subject because it's a dying subject. There aren't many taxonomists. And you can have links with museums in different parts of the country so that the taxonomic strength will increase, which will help you to identify the local fauna of your country, of your part of the country, of your state, of your region. This can be a beautiful topic again for thesis. Now, this is one example of how when an invasive enters the country, you can intercept it at the port of entry itself. Now, this was a consignment of rambutan which entered and came to the Bangalore airport. Now, can you see some white specks on the rambutan fruit? Now, this was found as a doubtful consignment. They saw these white specks and they sent it to NBAIR. Now, we have a mealybug expert, Dr. Sunil Joshi. He could identify this as a new mealybug, that is Excelomoculus philippinensis, which is an invasive pest. If this consignment had by chance entered our country, it would have destroyed 20,000 hectares of our sapota orchard. So it's because we had taxonomic knowledge to identify this invasive pest. It was not allowed to enter into the country and it was intercepted and blocked at the port of entry. Now, biological control approaches. I will just go to the basics a bit. As VC sir and Dr. Dilip Singh mentioned, there are three common approaches. One is the conservation approach. In conservation approach, what we primarily do is we advise the farmers not to use chemical insecticides so that what are the good bioagents which are existing in nature can be conserved. They won't be killed and they are allowed to perform and kill the pest. Or you can have crop habitat diversity to encourage the biocontrol agents, and you can have certain crops which will allow the pests to go away and which will attract the natural enemies. The second approach is the classical biological control, which is generally used when you have to tackle an invasive pest. And here, one of the common approaches is to import the natural enemy, the biocontrol agent, also from the country of origin of the pest, so that it can attack the pest and control it. Now, I'll speak more about it as we go on. And augmentation biocontrol is when we know that there are some biocontrol agents in nature, but it is not in sufficient numbers. In such a situation, you have to multiply the biocontrol agents and release them in the field as you do for chemical pesticides. You know, you don't expect one spray of chemical pesticide to do everything. So in a similar manner, in augmentation biocontrol, we can use parasitoids, we can use predators, or you can use pathogens depending on the stage of the pest which you are tackling. Now, there are some conservation strategies which help biological control. I am going through this primarily because VC sir also mentioned how biocontrol can be integrated with other approaches. So conservation strategies, basically, I am focusing on two important points. One is judicious use of insecticides and one is habitat manipulation. So I'll explain this with some examples. This is an example in rice, a work done by Indian Institute of Rice Research at Hyderabad. So here, to control the pests of paddy, what they are doing is they are having some floral diversity, which attracts the natural enemies, and they are focusing on having some pheromone traps to monitor the occurrence of the pest, and judicious use of insecticides. Insecticides to be used only when it is required, not to destroy the natural enemies, only when the pest is not manageable by the natural enemies. Now, this was a beautiful ecological engineering study where Dr. Chitra found that by planting marigold in the bunds, it attracted the natural enemies of the hoppers, the brown plant hoppers attacking the rice, and it reduced the pest population and thus improved the yield. Now, ecological engineering, why it is beneficial, I told you, by growing these flower buds, there is a higher floral diversity through which the parasitoids which are accumulating on these floral crops, their fitness is better, they become more, the survival is better, higher parasitism, and in the process, the pest is reduced, and eventually there is a real reduction in insecticide usage. So it's a holistic approach of managing insect pests by encouraging the biocontrol agents. So this is an integrative approach. Now, this is another example of habitat management. I think you know about a recent pest, an invasive pest which entered our country called the Rugo spiraling whitefly. And this is a parasitoid which is very good for controlling it. Now, what was found by Dr. Selvaraj of NBAIR was that if canna or banana is planted between the coconut trees as banker plants, it attracts the parasitoid and thus the pesticide use can be reduced. Similarly, when we talk of a recent pest, the fall armyworm on maize, in Africa, the strategy which they approached for controlling the fall armyworm, one of the strategies was, it is called the push-pull strategy. That is, they grew Desmodium as intercrop, and they grew Brachiaria as an border crop for maize. So maize is the crop. Instead of growing it as a sole crop, they had an intercrop of Desmodium and a border crop of Brachiaria. By doing this, the pest was repelled, pushed, and the natural enemies were attracted to the crop, pulled. So this is called a push-pull strategy. A similar study, I'm very happy to say, Dr. Amala conducted in NBAIR for fall armyworm, the first study in India of its kind, where she used a legume and maize intercropping, and in the border, instead of Brachiaria, which she could not get in Indian situations, she used Napier grass along the borders, and she found that natural enemy abundance was more, pest population was less, and yield could be increased. A very beautiful example of the habitat or the landscape on biological control. Here, you know what was done here was that this was done in USA. I am giving you some global examples, especially very recent studies, so that students have the inclination to go through literature, global literature, to understand what is happening in biocontrol in other countries versus what is happening in India. Here in winter grass, there is a very notorious pest which is called wheat stem sawfly. Now, when they were checking if there is any parasitoid of wheat stem sawfly, they found that absolutely zero parasitism in 30,000 stems which they sampled. But interestingly, what they found was there was a non-crop plant called Agropyron christistum. On this, they got Brecon parasitism up to 83%. So the understanding is, if you allow non-cultivated grasses to grow around the field, the Brecon parasite can move from the non-crop plant into the main crop when the pest has to be managed. So this is a wonderful example of, as such, landscape, how it is conserved to manage the pest. Now, promise of augmentation biocontrol. I spoke about conservation. I'm moving on to augmentation biocontrol. Now, in augmentation biocontrol, we can use parasitoids and predators, which are called macrobials, or we can use virus, fungus, Bt, entomopathogenic nematode, which is microbials. Now, in NBAIR, I told you we have the largest repository of both parasitoids, predators, and microbials. We have around 130 macrobials, including host insects, parasitoid predators, and we had around 623 isolates of fungus, Bt, virus, fungal pathogens, and EPL. Now, for augmentation biocontrol, there has to be a repository which can supply the biocontrol agents to release into the field for pest management. Now, this is the most common example. You all will be knowing about trichogramma. What I'm trying to give you here is some new approaches in trichogramma usage. So, as you all know, trichogramma egg parasitoids are produced using the rice moth, and we have a semi-mechanized system of producing trichogramma using Coursera cephalonica. And the Coursera production is kind of semi-mechanized. And using the Coursera eggs, we prepare trichocards. And these trichocards with the parasitized trichogramma eggs are cut into bits, and these are fixed on the crop. What is expected is the trichogramma adults are supposed to come out of these eggs, go in search of the pest eggs, and lay their eggs within the pest eggs and control them. So this is the science behind it. Now, there are different species and strains of trichogramma. I have given you some pictures: Japonicum, Embryophagus, Pretiosum, Chilonis, Bactriae, Echiae. Now, some of them are imported. They are not indigenous, and some are indigenous. Chilonis is indigenous. Now, this is Telenomus remus, a parasitoid which we had imported long back for Spodoptera litura management. We have this at NBAIR maintained. Now, these are some of the egg parasitoids. I'll speak more about it as we go on. Now, trichogrammatids can be used for management of various lepidopteran borers. For example, the paddy leaf folder, the bendy fruit borer, the paddy stem borer. We can use it against the brinjal shoot and fruit borer. We can use trichocards against the diamondback moth, which can attack cabbage and cauliflower. And we can use it even against Tuta absoluta, which is the tomato fruit borer. Now, this is how the trichocards have to be fixed on to the plants where we are trying to manage the pest. Now, I told you, various species of trichogramma, and we have a celionid egg parasitoid called Telenomus remus. Now, we have a strategy on which crop it can be used. You cannot use the same species for all the pests, for all the crops. So there is a tabulated information available. Since the topic is very broad, I'm just touching the important points. Later, you can check our websites for more information. Now, also, you cannot release as per will. There are fixed dosages when you have to release trichogramma. For example, if you have to release in mulberry against the leaf folder, you have to release 4 lakhs per hectare in 2 releases. And I should tell you, 5 cards make about 1 lakh release. So it's not a very huge number. The number of trichogramma, when we say 1 lakh or 2 lakh, 1 card contains 18,000 parasitoids. So trichogramma is a very minute parasitoid. So we need not get overwhelmed by the fact that you have to release 1 lakh or 3 lakhs. It is just a number based on the number in a tricho card. Okay. Now, primarily what I want to emphasize here to our MPUAT partners of AICRP biocontrol, to biocontrol researchers and others, we have modules in biocontrol, but the point is how we convince the farmers. We have to work with the farmers. We have to go and show them. It's not that we go to the university field and do our trials. We should do our trials in farmers field so that at least they are convinced about how it works, because it's not an immediate kill like chemical pesticides. You have to show them that after releasing, after so many days, how the pest population is gradually reducing. Now, this was the first case of a trichogramma production in the on-farm level, which was done by my colleague, Dr. Lalita. This is production of trichogramma on Eri silkworm eggs. So Eri silkworm, these are the eggs, the early instar larvae, the mature larvae of Eri silkworm, the cocoons and the moths. Now on Eri silkworm eggs, if we process it by storing them and putting them in KOH solution, there is a fixed protocol. Once you do that, trichogramma can parasitize. Now the advantage, 1 card of trichogramma using Eri silkworm eggs, you get much more trichogramma adults from 1 card. Reason is the Eri silkworm egg is much bigger. So from 1 egg, you can get 29 parasitoids, whereas from 1 Coursera egg, you can get only 2 to 3 parasitoids. So that explains how you can produce more. Also, another advantage, this can be produced in the farm level itself. This is a beautiful example of how a physically disabled girl, Sushmita, we taught her how to produce silkworm, Eri silkworm, how to use the eggs to produce trichocards. And this young lady, who was not contributing at all to her family, could produce trichocards using Eri silkworm eggs, and she could give it to her father, who's a sugarcane farmer, for controlling the sugarcane pest. Now, there is an example of augmentation biocontrol in the greater Mekong sub-region. Now here, it was a joint venture by IRI, Philippines, the Tiani Biocontrol Company, Switzerland CABI, China, Malaysia Institute of Plant Protection, Chinese Academy of Agriculture Sciences. So many organizations came together. They provided funding, and in the greater Mekong sub-region, 11 trichogramma rearing units were established. Now, this is the need in our country. Now, to some extent, we have succeeded in Kerala, where the group farming concept is there. Many farmers come together and adopt the biocontrol module. So they need supply of biocontrol agents. This was done by the State Biocontrol Lab in Kerala. They were supported by NBAIR by giving them good quality trichocards to start their culture. But this whole process they could cover 75,000 hectares of coal lands in 5 years. Now, this happened without any specialized funding. But you remember in the previous slide, in Greater Mekong sub-region, the amount of funding they received. But in our country, without that much of funding, we could achieve this. Now, with funding, what would be our achievement is what I want you to imagine. Another success story in rice is done in Andhra Pradesh, that is the Anakapalli region. She is Dr. Vishalakshi, one of our colleagues. What she did was she taught the tribal farmers the importance of trichocards. I was overwhelmed when I went there. One of the tribal farmers, he explained the use of trichocards by demonstrating the production in front of the other tribal farmers. Because instead of we teaching, when the tribal farmer explained to the other tribal farmers about the use of trichocards, they were so much more convinced. Now, moving on from egg parasitoid, I want to just give you an example of larval parasitoid, which attacks the larval stage. The best example is the coconut black-headed caterpillar, Opicina arenosella, and this is the kind of damage it creates. It is terrible. And this is the parasitoid which can be used to control Goniosis nephantidis. There is a production protocol available. And what is done is the cocoons of Goniosis is packed and sent to the field. And the parasitoid adults need not be released at the crown of the tree, which is very difficult. So what you have to do is you can just release it at the base of the tree. The parasitoid moves up and reaches the crown. And it was really a real good control of the Opicina. And this is how the damage was controlled. All the fresh fronds of the coconut tree were free of the damage by Opicina. You can see these are the old fronds which were damaged. The fresh fronds where it was released was free of damage. Now, to reply to one of the queries by Honorable VC, in some cases, biocontrol works as a standalone technology, but in some cases, you have to integrate it with various other technology. This is one example in CPCRI Kasaragod, Kerala, where for the management of the coconut black-headed caterpillar, they used the release of stage-specific parasitoids, and within a period of 2 to 3 years, what they found was that, see, what they did was they didn't use only Goniosis, they used Brecon also, and they tried in various states also. And what they found was that there was a damage reduction to 95%. Similarly, in the same institute, for managing the rhinoceros beetle, all of you must have read about the rhinoceros beetle. This is a kind of damage it creates in coconut tree. Now, they had different approaches combined for the management of Oryctes rhinoceros. They did leaf axil filling with botanical cakes. They used metarhizium for managing the larval stage. They used pheromone traps. So it was an integrated approach. And through this, they could get about 81% reduction. So depending on the case, it has to be integrated. Now, if we move on to the predators, I'm giving you a single example of ladybird beetles. This is Cryptolemus montrosieri, and this is Skimnus cacoxivora. Now, Cryptolemus is an exotic predator which was imported several decades ago, but it has established in our country now. And Skimnus is an indigenous predator. Now, the larval and adult stages of these predators can feed on mealybugs. See, these are all the mealybugs which are attacking different fruit crops. Now, this technology is available with many organizations, including NBAIR. So Cryptolemus and Skimnus can be produced and released, but you need large numbers. So here the challenge is for commercial units to come forward to produce Cryptolemus. Now here again, you have to take care of the release rate. If you are releasing in an agriculture crop, it should be around 5,000 beetles per hectare. If you're using it in a grapevine, it should be 1,500 to 2,000 beetles in a vineyard. And if it is a tree crop, it should be 10 to 12 beetles per tree. Now, this is an example where you may use two kinds of bioagents in the same place. Now, here, there is an integration of trichogramma in the brinjal field for managing the shoot borer, the fruit and shoot borer. And you can use Cryptolemus for managing the brinjal mealybug. Now, it is not necessary that only one agent can be used. Depending on the situation, you can use two kinds of agents. I want to shift your focus now to polyhouse pests. Now, many crops are grown in polyhouses, but thrips and mites cause huge problems in polyhouses. You can have swift-tooth thrips dorsalis or Franklinella schultzi, or the red spider mites, which can cause acute webbing, or there may be broad mites. Now, in foreign countries, I want to tell you, they are producing commercially predatory mites and predatory anthocorids. Both these are used in polyhouses. This is a predatory mite and this is a predatory anthocorid. These are used and sold in packets to commercial polyhouse growers. Now, do we have any such technology? Yes. This was a technology developed by me when I was a young scientist at NBAIR. This is an anthocorid predator. This is just to show you how an anthocorid predator can attack a tetranakid mite, and see, these are the healthy mites, but after its attack, the fate of the tetranakid mites. Now, like this anthocorid predator, this is called Blactostethys paliscens. I had 10 other species of anthocorid predators, which are still being maintained at NBAIR. This is another technology of producing predatory mites in India. Now, earlier, predatory mites were reared in net houses because they had to be reared on pest mites. Now, predatory mite rearing on pest mite, but pest mite you have to multiply in potted plants kept in net houses. Now, I developed a technology where I started multiplying one species of predatory mite, which is called Nucellus indicus, on a storage mite. So I didn't require a net house. I didn't require potted plants. I developed a simple protocol for multiplying predatory mite, one species. Now we can try it on other species also. And this technology, I'm happy to tell you, in the next issue of Current Science, this will be published. Now, what are the challenges for utilizing parasitoids and predators for biological control? The first one is availability. Where do you find it? People come from all over the country to NBAIR. The second challenge is how long we can store them. We should develop storage technologies for biocontrol agents. Now, how do the bioagents face abiotic stresses, chemical sprays and high temperatures? How do they manage? Now, do different biocontrol agents, are they compatible when we are releasing trichogramma? We are releasing a predator also. People think the predator will attack the trichogramma and kill. So are bioagents compatible? Now, in other countries, there are wonderful, huge commercial producers of biocontrol agents. Example, Copper Biological Systems in Netherlands, BioBee in Israel. But in our country, what are we depending on? We have a huge AICRP biological center project, biocontrol project, with centers in 29 centers in different parts of the country. I keep urging our centers to produce biocontrol agents, at least what is required for that region, so that biocontrol agents become available regionally. Only then the farmers or the stakeholders will be convinced. If for everything they have to come to NBAIR Bangalore, or if for everything they have to go to another city, the farmers or the other stakeholders will not be convinced. So AICRP biocontrol is our potential promise for producing biocontrol agents. An example is the State Biocontrol Lab in Mannuthi in Kerala. With just a small number of staff members, they are producing around 1,000 trichocards per day and around 25 tons of entomofungal pathogens. And this is another example of a farmer. He is actually an engineer turned farmer. He has devised a new technology for producing trichocards. See the innovativeness of a farmer. He has understood the importance of biocontrol and he keeps interacting with me even now. His name is Mr. Gopi Krishna, a wonderful entrepreneur. Now, storage technology. I said that is another challenge. I'm very happy to tell you, trichogramma, generally commercial companies are telling we won't take up trichogramma production because when the trichocards are ready, we are not able to sell them and we are not able to store and the cards are getting wasted. So my student and me, under a DBT project, we developed a technology by inducing diapause in trichogramma. This is published in Canadian Entomologist, which I have put here. And by this, it could be stored for 95 days. Trichogramma could be stored, trichocards, for 3 months. This is what I want to tell you. Now, regarding abiotic stresses, I mentioned to you that is another problem for macrobials. NBAIR has developed a multiple pesticide tolerance strain, which was evaluated in different parts of the country, including Rajasthan. There is a strain of trichogramma which can tolerate temperature up to 38 degrees centigrade. And there is another trichogramma which has been developed for higher searching ability, because people keep asking, when you are multiplying in small containers, in small packets, in small cages, when you release them in the field, are they able to search? So we have developed a technology by which a high post searching ability trichogramma has been developed. Now this has been widely evaluated by Dr. Jalali and his team. And regarding compatibility between parasitoids and predators, generally there is a feeling that the predators are feeding on trichogramma parasitized eggs. So there is no use of releasing trichogramma and predator together. So along with my students and my junior scientists, we had come up with a few studies in which we found that the parasitized eggs by trichogramma is not touched by predator. Predator doesn't want to feed on parasitized eggs of the pest, whereas unparasitized eggs they will feed. So you can see the comparison. That means they are complementary. You can release trichogramma and predator together because the predator does not like to feed on parasitized eggs of trichogramma. Now, how do we tackle invasives? In the last 10 years, 14 invasives have entered our country. How do we tackle them? One approach is through classical biological control. Now, this is a global example. Maize stem borer is a major problem in Africa. Now, what they had done is they had brought in Cotacea flavipes, a parasitoid, from different parts of the globe to Africa and released against Chylopatulus, which is the maize stem borer. Now, it started in 1968 and it didn't establish. Then they imported from Pakistan in 1991. Then they imported from India. From India, a lot of Cotacea flavipes was sent to Africa. And it was released right from 1996 up to 2003. Now, there are some interesting research questions. I want to leave it to the researchers. In 2005, when they checked all these released plots, they found that the parasitoid has established in all the countries except in one country. Why? Eritrea, that is one country where it didn't establish. Was there any indigenous natural enemy there, which was not allowing the exotic natural enemy to establish? Now, Ethiopia is a country where it was not released at all, but there the parasitoid established. Why? So these are certain interesting research questions. Now, the Pakistan population, only one population was released in Kenya coast. It established. Whereas in Zimbabwe and Zambia, several populations from different countries were released. They also established. So what is the reason? So is it important to release a single population, or is it important to have a genetic diversity and release large population for controlling a pest? All are research questions. Now, the best example of tackling an invasive through classical biological control is the papaya mealybug example, which I keep repeating because it indicates how a single parasitoid, Aerophagus papayae, managed to control the papaya mealybug in different parts of the country. But here again is a question. Along with Aerophagus papayae, two other parasitoids, Anagyrus loci and Pseudoleptomastix mexicana, were imported. But these two did not establish at that time when we initially released in the field. But over the period, my colleagues at NBR are saying, now in some parts of the country, like Salem, Pseudoleptomastix mexicana is emerging. What is the reason? Over the period, is there a difference in the parasite populations? The management of the weeds through classical biological control. This is an example where the Salvinia weed, Cytobagus salviniae, was controlled using Cytobagus salviniae is the weed insect which was used for controlling Salvinia molesta. You can see here, this is the weed logged area and this is the free area after the release of this weevil, which is the weed insect. It was imported from another country. But now water hyacinth has replaced Salvinia. What is the reason? Some questions. Now the classical biological control of some of the pests This was an example in outside India. The rhinoceros beetle was controlled using virus, that is the Oryctis rhinoceros nucleopolyhedrosis virus. But over the period, what happened? That in some regions, for example, in Guam, it was not getting controlled. Why? The Guam population of the rhinoceros beetle was tolerant to the virus. So these are the reasons why biocontrol does not work sometimes. There are some strainal variations in the pest which prevents the biocontrol agent from functioning. Now, how do we tackle invasives through conservation biocontrol? The best example is the management of sugarcane woolly aphid. Here, what we did was we just told the farmers not to use chemical insecticides. By doing that, 3 natural enemies got conserved. That is Encarsia flavoscutellum, this parasitoid, and 2 predators, Difa and Micromus. And this brought down the population of woolly aphid in different parts of the country. Now, this is another example of spiraling whitefly Eleorodicus dispersus. The parasitoid for this particular pest was brought from the Lakshadweep Islands, this parasitoid. It is called Encarsia guadeloupe. This was brought into our country for managing Eleorodicus dispersus in 1995. Keep this in mind. But recently, when the Rugo spiraling whitefly started attacking, the same parasitoid which had come for the spiraling whitefly started attacking the Rugo spiraling whitefly also. So conservation was the only strategy here to conserve this parasitoid to allow it to control the Rugo spiraling whitefly. Now, how do we tackle invasives through augmentation biocontrol? So for invasives, it is not only classical biocontrol, it is not only conservation biocontrol, it can be augmentation biocontrol also. Again, one example in Alberta, there was a notorious pest which is called amber-marked birch leaf miner. For this, several native natural enemies like Aptesis cygnus, Lathrolestis operi, all these local natural enemies came together and started controlling this invasive pest. And for us, the significant example is the entry of the recent fall armyworm Spodoptera frugiperda. As you know, it attacks mainly maize, sorghum, and bajra. There are, of course, instances where it is attacking other crop pests, but these are minor examples. The major host plants are maize, sorghum, and bajra. And this is the kind of damage fall armyworm creates, which really causes terror in the farmers. And here, these are all the natural enemies which we found in nature. Now, our job was only to see that these are conserved. Out of these natural enemies, which is there in our country, which could attack fall armyworm, these two, Trichogramma chilonis and Telenomus remus, we are able to multiply in large numbers and augment. So we can use this to target the egg stage of fall armyworm. Besides, in the field itself, we could find Nomuria rilei attacking the larval stage. We could find virus, the Spodoptera frugiperda nucleopolyhedrosis virus attacking. So at the end of this, what was done at NBIR was a holistic bio-intensive IPM module. Without chemical insecticides, we used other approaches. What we did was a pheromone for the initial monitoring of the pest or for mass trapping. Egg parasitoids, we have chilonis, we have Tritiosum and Telenomus, one of them to attack the egg stage. And for the larval stage, we have our own isolate of Metarhizium, we have our own isolate of BT, we have our own isolate of virus, Spodoptera frugiperda NPV, and we have entomopathogenic nematodes to tackle this pest at different stages. Now, this was done into a module and we tested this in farmers' fields to convince not only the farmer, to convince ourselves that these work. But there are problems when we are using microbial insecticides because they have to be registered before we can use it in the commercial way. So that is the thing which is remaining with us. Now, microbials, I told you, BT can be used, fungus can be used, virus can be used, but entomopathogenic nematode, I want to focus a bit because of its advantage. One is it can be produced on Galeria larvae, and the earlier technology was to multiply on Galeria larvae and using the Galeria larvae for release in the field where we have to target the pest. Now, even there is a beautiful reference which I read yesterday. This is a 2021 reference where they speak that there is a very close evolutionary relationship between the entomopathogenic nematode and grubs, the white grubs or the root grubs. And another advantage of entomopathogenic nematode, this is another beautiful study in 2016, where they released entomopathogenic nematode to control Spodoptera exigua. But while using entomopathogenic nematode, it not only controlled Spodoptera exigua, it also made the plant resistant to Bemisia and Pseudomonas disease. So it had a multi-pronged approach. That is the advantage of using entomopathogenic nematode. I told you it has a close association with white grub because it is the best agent to tackle the white grubs. Now at NBAIR, instead of using the Galleria larvae for releasing, NBR green technology has been developed. That is a vegetable powder formulation which can be used for controlling, and this has been widely tested in the cardamom fields, sugarcane fields, areca nut fields, and it has been commercialized also. Companies have started producing, and how? Because this does not need registration. If there are entrepreneurs in the audience, I would urge you all to take up this technology from NBAIR, because this technology can be mass produced, EPN can be mass produced and sold. It does not require registration from CIBRC because registration is a very cumbersome process. Now, what are the recent challenges we are facing? One of the most recent one is the locust. Now, locust was also tackled using biocontrol in Africa. What did they do? They used a fungus which is called the green mussel, and this was mass produced, and it is actually a variety of Metarhizium anisopliae. It is variety Acridium, and this was released using drones or even ultra low volume spraying through aerial methods. Now, after using the Metarhizium, see, this is the infection in the hoppers, that is the locusts in Africa. Now, this project was called the Lubilosa project. I want to tell you about this because it took 13 years from November 1989 to 2002, from isolation of Metarhizium acridium till commercialization, and so many organizations, millions of dollars were given by organizations. Can we expect this kind of a funding for Indian initiatives to control these notorious pests? In India, this is work from NBAIR. There are two isolates which appear to be very promising for locusts. This is by Dr. Ramanujam, one Beauveria isolate and one Metarhizium. And we had urged our Udaipur Center at MPUAT, Ludhiana Center and Anand Center to evaluate these strains against locusts in their respective states, because we cannot bring locusts into our country, into our part of the state. Now, this is another recent pest which has entered our country, the cassava mealybug, for which we need a parasitoid to be imported, which is called Apoenagyrus lopezi. Why I am telling all this? Again, for Parthenium to control Zygogramma bicolorata was a weed insect which was imported long ago. But there is a problem here because Zygogramma can feed only on the leaves. It cannot feed on the seeds and flowers. We need to import another parasitoid, another weed insect, which is called Smicronix lutulentus. So to import, we need collaboration with CSIRO Australia. Already CSIRO Australia has promised. Dr. Dileepan is giving us the cultures. Now, in the cassava mealybug example, which I told you over here, and the example of Zygogramma, we need cultures from other countries. So what is required? We need collaboration. We need in biocontrol, if we have to succeed through classical biocontrol, we should be able to import biocontrol agents from other countries. So importance of networking, I want to emphasize. This is an example in Australia where the red crabs were a great tourist attraction in the Christmas Island National Park in Australia. And this ant, which is the crazy ant, was a problem killing these red crabs. So what they did was This ant had an association with the scale. So what Australian researchers did, they imported a parasitoid which is called Tecadiphagus somervillei from Malaysia to control the scale, and the scale was killed, by which the ant could not have the mutualistic association. The ant was also killed and the red crabs were saved. So to do this, it was a multi-organizational, multi-country networking. Only then this biocontrol succeeded. So in our country, if we have to import, we need permission from DPPQ&S. If we have to help another country, we want to exchange. We want to send our biocontrol agent to Australia, for example, and get a parasitoid from them. We need permission from National Biodiversity Authority. So the problem is that there are delays in getting approvals and permits. So the challenge actually lies in streamlining these procedures so that we can get our approvals earlier, so that we can help each other, so that the global farmer is helped. We are not thinking of Indian farmer alone through biocontrol. Now, first and foremost, there should be systematic monitoring for invasives at the entry points. There is a clear dearth of taxonomists. I urge the researchers who are all here to focus on taxonomy among students. There is a clear scarcity of funds. I urge young students and researchers to apply for biocontrol based projects to DST and other funding agencies. I assure you, you're going to get it. Try to think of network projects internationally and nationally. And here again, I urge students to apply for funding to present papers abroad. See, now everything is online. You can present your work and you're going to get projection. At the same time, you will learn about what is happening in other countries. Regulations, which I told you is the major constraint, registration of our biopesticides. We need streamlining so that the biopesticides which we are producing in our country gets registered, only then the farmers will get. So the national and international networking, I should say, is key for strengthening biological control in India. So the take-home message today, classical biological control we should take up only if after evaluating our indigenous natural enemies, if nothing is available, if no isolates are available locally, only then think of classical biological control. You can use two natural enemies by checking if they are complementary. Introduce natural enemies may enhance the effectiveness of local natural enemies. It is not that, there is a myth, as Honorable VC said, that it will cause harm. It is not necessary. And another thing I want to tell you, if we are bringing an exotic natural enemy, we are not directly putting them in the field. There is a quarantine procedure where we test against non-targets. Only if found 100% safe, they are put into the ecosystem. Resolving the taxonomy of pest and natural enemies, the relationship between biocontrol and taxonomy. Please take up taxonomy related projects for your MSc and PhD. We have to have perfect planning in choosing a biocontrol agent. There should be national and international networking. And of course, there could be unexpected failures, that could be unexpected successes. Now, I have given you a lot of success stories, whatever I could think of within the time frame allowed. But then if we take up more projects, more networking, there is an opportunity for all of us to speak about more success stories. Thank you all for your patient hearing. And this is one of the favorite pictures that I have where I'm with my colleagues for our last foundation day and my institute, to which I'm always ever grateful. And thank you all, and be safe, and thank you once again.
Thank you, madam, so much for your wonderful lecture, which was backed up by a very vivid and a very lively, evocative presentation as well. And I think it was so simply drafted that all the students must have really been benefited by it. And your last couple of slides, they really have touched us. It's not all about rosy. It's also there are many challenges before us, and what message we should really take home, how we can collaborate, how we can cooperate, and how we can create a sort of networking for the biological pest management. Thank you very much, madam, for your
Thank you, sir.
kind
Thank
lecture.
you, sir.
Ladies and gentlemen, I would now request our Director of Research, Dr. S.K. Sharma Saab, to recapitulate some of the important points that Madam has raised and to wrap up all the technical things about biological pest management. So over to you, Dr. Sharma, sir.
I'm
Thank you,
sorry, Dr.
Dr.
Nepalia, if I have overrun my time. I'm so sorry.
Not at all, madam. It was treat listening to you, and I think would have even loved if you could have taken a little more time. I
I'm
think
really
there's no
sorry
issue.
because the topic is so huge. I could not restrict
Yes, yes, yes.
myself to.
No, mind, madam. It was a great lecture. Thank you, madam.
Thank you.
Thank you, Dr. Virendraji Nepalia, sir, our Honorable Vice Chancellor, Dr. Narendra Singh Rathod, our esteemed speaker for today's webinar, Dr. Chandis Balal, Madam from the Bangalore, our respected Dean, Dr. Dilip Singh, Dr. Ajay Sharma sir, Dr. Jingarji, and Dr. Mini Srivastava Madam, the Organizing Secretary, and the Head of the Department, Professor M.K. Melaji, all organizing team, our senior faculty members, Registrar, Comptroller, and all the participants from different states of the country. It's really a matter of great privilege and proud for me to be the part of this wonderful webinar on a topic which is the demand of the 21st century. And I think the well tone was set by our Dr. Vendraji Nepalia Sahab in the introduction itself about the activities of the MPUAT and the relevance of the topic of the webinar, but it was wonderfully mentioned as a background by our Dr. Dilip Singh, the Dean RCA, who rightly pointed about that there is a need of the pest control for the food security of the country, but at the same time, I have to minimize the use of the chemicals. It is very important moment for the not only at the national level but at the international level. So we have to switch over to the IPM strategies and biorational pest management strategies. Later on, our chief guest and the patron, our Honorable Vice Chancellor, Dr. Narendra Singh Rathod Sahab, who covered right from A to Z of the integrated pest management as well as the principles of the pest management, and he emphasized well that food security is the main concern and use of the chemical is very important. But at the same time, protection of the crops from the right methods is also important with respect to the environmental concerns as well as the food security, because nowadays people are giving more concern, and this concern, as we know, that also pointed out in the historical aspect in the presentation of our eminent speaker, that with the publication of the book by the Rachel Carson in 1962, the Environmental Protection Authority was established in the US, and later on every project, whatever it may be the agriculture or industrial project, they have been related with the environmental concern. And I think our eminent speaker, Madam Balal, she has wonderfully taken up all the topics with, the talk was very informative, and it shows that throughout the life career, Madam has absorbed all issues with his hard work and intelligence. And it was flowing with his humorous words, and it was so simplified presentation that though I am working since last 25 years in the area of organic farming and sustainable agriculture, but I have not seen such a wonderful presentation on the biological control and how to specifically that with respect to the several examples at the national and international in the area of the bio control agents. And as our Honorable Vice Chancellor mentioned, that for the maintenance of the quality production, a mediocre policy where how to integrate the use of the biopesticides and biocontrol agent is very important, though he also mentioned about his concern about the soil and water pollution and degrading soil fertility as well as other aspects of the sustainability. But he also mentioned the different methods of the pest control, where biological pest control, its boundaries as well as its scale is not increasing in our India. That was the main concern of our Honorable Vice Chancellor, where he emphasize that how this biological control can be expanded at the farmers level so that it may be more remunerative, it can reduce the cost of production, it can enhance the productivity, and at the same time, a quality food can also be provided to the emerging population, but wonderfully taking into the subject as well as the in-depth analysis of the topic, that how sustainable pest management is more important for our country. It was given by our eminent speaker, respected Madam Balal, and she nicely introduced the topic by giving national and global examples, where he emphasized that more and more number of such projects should be taken up by our students and researchers. And as in the last slide, she also mentioned the challenges, that there is a dearth of such researchers and taxonomists who are interested in taking up such more number of the biological projects, so that this discipline can more be promoted, if such kind of the more projects would be there, its expansion at the field level would be more applicable. And she also emphasized as a take-home message that more number of entrepreneurs should enter into the area of the biological control agents. And as being the very eminent expert in the area of the biological control and in the entomology itself, he emphasized all principles of the sustainable pest management technology, where she very clearly highlighted that we cannot completely ban the use of the chemicals, but we have to reduce the use of chemicals by integrating more and more use of the different bioagents, whether it may be predators, it may be the parasitoids, it may be the parasites, and with the three different classical approaches, wonderful examples from the different parts of the country, mostly from the Kerala and South, and some part of in covering all crops. Either it may be the fall armyworm, the maize, it may be the sugarcane, it may be banana, it may be rice. And she wonderfully give the how the on-farm production of the different this trichogramma can also be done. It was really a fantastic example that we can promote such kind of the activity, so that at the lesser cost of production, this can be permitted at the farmer's field. And it is also one of the wonderful examples for our students, that they can also learn under the student ready program such kind of the activities. And good examples were also quoted by the madam for the control of the thrips and the mites in the polyhouse, which is the urgent need of the present era. Many farmers and many polyhouse growers are searching non-chemical pest management nowadays. Even we are also getting so many phone calls. Sir, without the chemical, how can we control the thrips and mites, especially in the use of the predatory anthocorid? I will also urge to our head of the department, other entomologists at our university, to kindly get in touch with the madam, and how can we take this technology and can popularize this technology in our area in the Rajasthan itself. And a number of interventions at the national level, like the Copert biological system in Netherlands, they are the classical examples of the biological control, because basically the biological control itself was started from the 1940s from the Europe and the US itself. So there are very good methods available in India. That madam herself has been at the helm of affairs as a director of the NBIR and the project coordinator of this All India Coordinated Project on Biological Control. And that is really all your experience was coming to the all slides and your presentation. Your voice was so wonderful, and technically you give so summarize this narration of all the topics, that really I came to know that how we can integrate the biological control into different crop pest management strategies. But at the same time, you also narrated that the issues of the long-term storage, I think we are also feeling at our university that after production of the trichogramma, even the chrysoperla and anthocorids, how they can be stored for a longer time. So the 90 days storage period by the technology developed by our centers, it is really a good kind of the information that we receive. Like the abiotic stress, this is one of the good scientific advancement that most of the participants learn, and we really got a specific confidence that there are many technologies available which we are not aware, and we are worried about these aspects, but such kind of the scientific advancement is available in our country and abroad. We can learn by having the contact with the good experts like you and other experts as told by you. And similarly, the aspects of the better searchability and compatibility was also told by you. And at the last, tackling invasiveness for the papaya mealybug, even this in case of the water hyacinths, and in case of the different beetle, and specifically the maize stem borer, then fall armyworm, and many examples. Really, they are very wonderful to learn, and this gives a confidence that biological control has a lot of successful stories. But we can avoid the failures also in the long term if we have the right strategy for its management at the lab level, at the policy level, at its registration level, as well as having the proper number of the projects, as told by you, that there is a really dearth of the fund. And I would say by my own experience that there is a dearth of the actual scientists who are working into the biological control. Though as such, the percentage of the biopesticides or biocontrol agents used in our country and abroad is not more than 6% of the total pesticide market. And I came to know in some of the literature that only 0.5% of the scientists, specifically the entomologists and taxonomists, they are in the field of the biocontrol agents, which also indicates that there is a need of specific emphasis on such kind of the subjects if you want to really have a concern on the sustainability of the food production, and a real emphasis on the on-farm application of the sustainable pest management technologies. So I really congratulate to our eminent speaker, madam, and our honorable vice chancellor. I express my specific gratitude for a very informative and wonderful talk, and it was really very caring the very in-depth information for all the students, researchers, our scientists from the different universities. And I expect that now onward, you will be getting a lot of phone calls from our sides to learn more things in this direction and to get your good direction, specifically from my university, because many times our Honorable Vice Chancellor, Dr. Narendra Singh Rathod Sahab, has taken your name and about laurels, about your eminency in the or expertise in the area of the biocontrol management. So I expect that all students and scientists have benefited a lot from your this very informative and wonderful talk. And I express my sincere gratitude and a big salute to you for your all care and share you have given to the MPUAT, and also to my Honorable Vice Chancellor, Dr. Narendra Singh Rathod, who has always been very wisdomful and very critical to take up such kind of the issues which are of international and national importance. And it is his searchability, I would rather say, that he has brought such kind of the experts with us. Thank you very much. And at the last, I congratulate to the Dr. Vendraji Nepalia sir and our organizing secretary, head of the department, Dr. Mehla sir, for a wonderful program and a number of participants who have connected with us. It shows that how important the topic, experts, and our organizers are. Thank you very much.
Thank you. Thank you so much.
Thank you, Dr. Sharma, for summarizing the proceedings and putting forth our views on the subject. Madam has already written in the chat box that if anybody has got any query on with regard to this subject, he or she may email it to Madam. And I think due to paucity of time, we may not be able to take up any questions right now. But please feel free to mail your questions to Madam. She has already communicated her email ID in the chat box. Thank you, Dr. Sharma. Now we wish to really come up towards the end of this online session, and may we request our head Department of Entomology, Dr. Manoj Kumar Mehla, to propose a vote of thanks. It's over to you, Dr. Mehla.
Good morning to all dignitaries and participants. Honorable Vice Chancellor, Dr. N.S. Rathod Sahab, MPUAT Udaipur. Respected Madam Dr. Chandis Balal, former Director, NBIR Bangalore. Our most valued guest, Dean RCA, Dr. Dilip Singh. Director Research, Dr. S.K. Sharma Sahab. Director Extension, Dr. S.L. Mundra Sahab. Dean CTI, Dr. Ajay Sharma Sahab. Dean College of Agriculture, Bhilwara, Dr. K.L. Jinger Sahab. Registrar Madam Kavita Pathak. Comptroller Mrs. Manjuwala Jain, and other associate members, participants, ladies and gentlemen. It's my great privilege to propose a vote of thanks to those who have directly and indirectly contributed to this webinar on promise of biological control for sustainable pest management, organized by the Department of Entomology, Rajasthan College of Agriculture, MPUAT, Udaipur. On the behalf of the Department of Entomology, the entire organizing team and fraternity of Rajasthan College of Agriculture here together, and on my behalf extend a very hearty vote of thanks to the guest speaker, Dr. Chandish Balal, for gracing and sharing with us on the experience of biological control for the sustainable crop production. I must mention our deep sense of appreciation for Dr. Chandish Balal, Madam, a highly reputed and esteemed personality in the field of biological control, and her contributions have brought about significant changes for her explanation of promise of biological control in sustainable pest management, and also to express our sincere thanks to Madam for giving excellent coverage and for providing encouragement on the today's topic. We are thankful to our Honorable Vice Chancellor, sir, for sharing his vast experience and immense knowledge and sparing his valuable time, support, encouragement, and guidance for the organization of this webinar. I also extend my thanks to Dr. Dilip Singh, Dean RCA, Dr. S.K. Sharma, Director Research, S.L. Mundra, Director Extension, Ajay Sharma ji, Dean CTI, Dr. K.L. Jingal, COE, and other university dignitaries for their enormous cooperation in the organization of this webinar under NAHEP component. Our heartfelt thanks to our colleagues, scientists, students from other institutes and universities for their active participation in this webinar. With these warm words and a kind message, we move to the end of today's webinar. Thank you very much once again. Thank you, sir. Thank you.
Thank you, Dr. Mehla. It was a really wonderful hour and a half with Madam, and I think we'll cherish the memories of this webinar for a very, very long time, and maybe that shortly we may again call you sometime. I think Dr. Mehla would be in touch with you, and maybe that you may have to once again come to us. Thank you very much, Madam.
Thank you so much.
Thank you,
I am
with the permission
honored.
of great, Madam, great. So nice of you. And
Thank
now
you
with the
so much.
permission of Honorable Vice Chancellor, we end the session here. And thank you very much for joining us. Thank you.
Thank you.